So I'd like you to start with introducing yourself. Let me know a little bit about what brought you to Colby, and maybe tell us a few sentences about the organization that you created.
My name is Jonathan Latham. I'm a PhD virologist who studied plant virologist, that is. And I did my PhD in England at a government institution that I was basically doing standard molecular biology, cutting and pasting DNA, finding out what effects that has on the virus that I was studying. And I did have conversations at the time about the safety and probability of things going wrong around the subject of genetic engineering. And they were interesting conversations. And then I had some other interesting conversations to do with finding out, for example, that the fantastic greenhouses that we had, that were the biosafety containment greenhouses, for where we were keeping our plants. So where my PhD was happening was in a government institute, which it was the biggest institute in the world for the study of genetic engineering of plants. And they built this super fancy set of containment greenhouses. They were negative pressure greenhouses, so that when you went into the greenhouse, all the air, when you opened the door, all the air would rush out from behind you, so that nothing could, flies or pollen or whatever it is, or genetically contaminated debris wouldn't leave the greenhouses, right? Because all these uncontained experiments are happening inside the greenhouses. And what I discovered from the biosafety institute biosafety person was that the greenhouse, nobody had ever thought about what happens to the water washings when the plants have been watered. What happens to the water that leaves the greenhouse? It was just going into the river, but nobody had thought about this, right? And this was really interesting to me because the whole concept of risk assessing GMOs is that you've got to think about a lot of different things. These are living organisms, and you have to think about their pollen, the fact that they reproduce by seeds, the fact that animals may come and eat them, that they may poop out that food, that there's a whole ton of ways in which genetic material and DNA can escape from a containment greenhouse. And they hadn't thought about one of even the most simple and basic ones. So they spent $5 million on these incredibly fancy greenhouses and totally failed to understand the basics of what was going on when you grow plants. And then we had conversations about the wisdom of putting bits of viral genes into plants, like what happens to when other viruses come and enter those plants. Will that DNA or RNA recombine? There were all sorts of biosafety issues, and colleagues would say things like, I don't like the experiment that so-and-so is doing because this is a particularly dangerous gene. We already know that. For example, like in this one case, it was a movement protein was being put into transgenic crops. And we know that the reasons why different viruses move in different crops is often because of their movement functions. So often, many, many different viruses, they have basically identical replication strategies. They have very similar ways of spreading through by insects. But what distinguishes why a virus can infect one plant and not another one is the fact that some can move through certain types of plants and some can move through other types of plants, and that those are the determinants in many cases of the infectivity of a virus. And so messing around with the movement proteins of viruses is especially a dangerous thing to do. And there were people at the time who were proposing to make plants resistant to viruses through making them transgenic with their movement proteins. So we had issues with those kinds of experiments. But at the time, I didn't realize how lax and badly thought out the commercial GMO regulations were. I mean, there was only one GMO had ever been on the market in Britain, which was the Flavor Saver tomato. I think it came out in 1988, and there were a few on the shelves. Consumers didn't like them, and they were taken off eventually after about 4 or 5 years. So for the most of my period of my PhD, which is 1991 to 1996, there were no GMOs on the market in Britain. And so we thought of them as abstract concepts, not as real things that people were going to put on the market, whereas that was actually starting in the US. By the time I was finishing up my PhD, there was a virus-resistant squash. There was the virus-resistant papaya was not on the market. There was Roundup-Ready soybeans that had already been approved in the US. And but I didn't think it was my business to do anything about these crops. This is what other people are doing. But I finally, I was confronted with a number of issues a little while later, because there were a lot of people who were, in Britain especially, who were concerned about GMOs, but who didn't have the expertise to put their concerns into context, to understand what they were being told by regulators or by company representatives or by government officials. And so they wanted my help. So people, when they found out that I was, had been a genetic engineer, they wanted to know my take on all this information. And so I ended up reading around some of the issues that, as a scientist, I probably wouldn't have bothered doing, and coming up with all kinds of facts that seemed to me rather interesting and pertinent, and that weren't being discussed by the regulators, by the company people, and government officials. So that's kind of my history. That was kind of my training ground of becoming a public interest scientist in a self-propelled kind of active way, as opposed to a sort of passive way of, I work for a university, or, I'm being paid by public money, therefore I must be a public interest scientist, and I don't need to take into account what's going on in the real world, as it were. And there were a whole series of things that already had alerted me to the fact that there were issues in and around the science of what I was doing. There were things like, for example, I didn't have a lot of respect for the people who often rose to the top of the profession. The people who had a lot of integrity, the people who did careful science, were not rising to the top and becoming institute heads and professors. It was the people who were gaming the system and interested in careerist aspects of science, right? So that was a red flag for me. Another thing that was a red flag was the fact that we were doing molecular biology in a lab in which we never visited the farmers, right? So we were doing all this research, and which we wrote and supposed and believed was for the benefit of farmers. But if you went far enough in your analysis, you could see that no farmer was demanding this research, and in fact that in many cases the people who are likely to benefit would be plant breeding companies, and often which would be multinational companies. There would be people who would who had agendas in other countries, for example, that were not necessarily beneficial to the people living in those countries. So there was a whole load of politics going on, and also the fact that everybody could see that this was research that worked for the benefit of the agricultural status quo, right? So what was really interesting was that there were researchers saying we should stop using pesticides. There was researchers saying we should have more sustainable methods of agriculture, that we should look after the soil better and such like. But that research seemed never to get done, right? Whereas all the research that we were being funded to do generously was to do molecular biology, which had a kind of uncertain prospects. And so that's also was a red flag. But I didn't necessarily put all these things together at the time. Like, it takes a while, as you develop your career, and things happen in sequence and sometimes years apart, that and some people, I think, never put all these things together. And it was partly leaving plant biology and becoming a nematode genetics researcher that enabled me to put these things together too. That it was a kind of a safe space to critique what I'd previously done, and then to see that it didn't fit together in a very wholesome way. So that was a whole process. It took probably 6 or 8 years for me, and in many ways it's still it's an ongoing development of my intellectual understanding of the place of agriculture in the world, the place of agriculture in the economy, the place of agriculture in politics, and there's so much to learn and so much information that's been hidden, suppressed, denied in all these stories, because these are the stories of poor people, these are the stories of the researchers who were ignored, these are the stories of the politicians who were ignored. So this is kind of, in a way, it's the anti-history, if you like, of society. And so it's hard to come across this information and synthesize it. But science and agriculture and politics, I've come to believe fit together in quite a tight web of synergy, which benefits certain groups in our society and is not of benefit to other groups. They're all part of accumulating wealth and denying people information and disempowering people, so that we don't have, the kind of society we want is in large part because we don't have the agriculture we want, and we don't have the science that we want, and we don't have the politics that we want. These things are kind of inseparable from each other. You can't think about them very well if you don't understand all the different parts. And so understanding the science for me has been a route into understanding these other issues. Because I don't have a background in agriculture, but I do have a background in ecology and understanding plant biology. But I didn't have a background in political analysis or public policy or any of those things, but I've started to understand public policy because of understanding the science.